Listen, it's my distinct pleasure to welcome Aspram Sargsyan. Student Dr. Sargsyan is familiar to me. She's been my advisor for the last 4 years, my advisee for the last 4 years. By way of introduction, Aspram came to KSU with a BA in molecular and cell biology with an emphasis in physiology from UC Berkeley. She also had studies in general medicine from Yerevan State Medical University in Armenia. At Berkeley, student Dr. Sargsyan was awarded 2 Science STEM awards and also one from the NSF and Los Rios STEM award. She was also Undergraduate Research Assistant in the lab of Dr. Jeremy Thorner, at which time she gained lab experience looking at septin proteins in yeast model Saccharomyces cerevisiae. This led Aspram to be a co-author on 2 articles in 2016 in very prestigious journals, including Molecular Biology of the Cell. She founded the Botany Club at American River College, was the VP of Armenian Student Association, has tutored and led sessions, and brings a vast list of volunteer experience. Moreover, she's interacted with the MKRC, this organization, through Dr. Rogers and Dr. McNab's studies that the MKRC is sponsoring. She's also engaged in preparing grant submissions and the publication of sterilization and sanitizing of 3D printed N95 masks that we did in this laboratory. So it's my pleasure. Let's everybody unveil and welcome our speaker. Say hello. Take it away, Aspram.
Hi, everyone. Thank you so much for coming today. The study that I will talk about today is titled An mRNA Vaccine Against SARS-CoV-2 Preliminary Report. This was published on July 14th in the New England Journal of Medicine, and it was co-developed by the researchers at the NIH and Moderna. The reason why I chose this study to talk about over all other vaccines developing right now in the world, coronavirus vaccines, is because this one is being talked about a lot in the US, and it almost sounds like this is going to be the one that might be administered sometime late, might be ready to be administered sometime late this year and early next year. However, that being said, there's a race of coronavirus vaccine development going on around the world. And before diving into the details of this trial, I will give an overall overview of vaccine development in general, and will briefly discuss some of the more promising trials going on right now in different parts of the world. So this slide is from the FDA website. It goes over how a new vaccine is developed, approved, and manufactured. So before entering the clinical phase, there's a clinical phase where the vaccine is being administered on animal subjects, and after it passes that phase, it enters the clinical trial, which consists of 3 phases. So phase 1 includes 20 to 100 healthy volunteers, and the main question being asked in this phase is this vaccine safe? Is it working? And are there any serious side effects associated with it? How is the size of the dose related to side effects? As it proceeds to phase 2, it involves more volunteers, several hundreds of them, and the questions are asked are, what are the most common short-term side effects, and how are the volunteers' immune systems responding to the vaccine? Phase 3 trials involve more volunteers, hundreds or thousands of volunteers, and the purpose of this phase is to compare people who do get the vaccine to people who get the placebo, and again, look at the safety profile and if it's effective or not, and what are the most common side effects. And after it gets approval, after the phase 3. It gets to the approval phase, and after it's approved by the FDA, they have to check each one of the lots that contain the vaccine, and they have to make sure that each of them are potent and safe and have high quality. And then after that, the FDA is supposed to inspect the manufacturing facilities regularly to ensure quality and safety. So right now, there is a number of trials going on in order to develop the perfect vaccine for coronavirus around the world. There's more than 155 vaccines against coronavirus currently, with 23 of them being in human clinical trials. This is a tracker from New York Times website, and if you click on each one of these, it will show the list of these trials with the titles, and if you click on them, you can see if there is any published results. You can take a look. If you're curious about this one approved one, it is by this company, by a Chinese company, CanSino Biologics. They developed a vaccine based on an adenovirus called AD5 in partnership with the Institute of Biology at the country's Academy of Military Medical Sciences. In May, they published promising results from phase 1 safety trial. Unpublished data from phase 2 trials demonstrated the vaccine produced a strong immune response, leading to Chinese military to approve it on June 25th. The company, however, won't say if this vaccine is going to be mandatory for soldiers or not. Another one that is promising, and Dr. Schneider shared this information, thank you for that, Dr. Schneider, is going on in Russia. I couldn't find any published results from this trial, but the researcher did give an interview to Medusa, and he stated that he administered the vaccine to himself and 76 other volunteers, each of whom had maintained a journal noting any side effects like fever, rash, irritation. And so far, fortunately, there haven't been any serious after effects. The researcher says that unlike the coronavirus vaccines now being developed in China, Great Britain, and the US, the vaccine that he's developed is a 2-vector vaccine, meaning that he uses 2 types of adenoviruses. So they administered 1 shot of human adenovirus type 26, and then 3 weeks later, another shot of human adenovirus type 5. And Lagunov told Medusa that his team has measured better immune responses to vaccines using different vectors, whereas booster shots that rely on the same vector sometimes lead to weaker immunity that fades after 3 to 6 months. So really promising, and hopefully soon we will see some published results available to the public. Another one that is being talked about a lot, and I feel like it will beat the Moderna, but who knows, is the vaccine developed by British-Swedish company AstraZeneca and the University of Oxford. This one is based on chimpanzee adenovirus. In their phase 1, phase 2 trial, which they published on July 20th in the journal Lancet, they found that the vaccine was safe, caused no side effects. It raised antibodies against the coronavirus as well as other immune defenses. The vaccine is now in combined phase 2, phase 3 trial in England, as well as phase 3 trial in Brazil, South Africa, and the project may deliver emergency vaccines as early as October. AstraZeneca has said that their total manufacturing capacity for the vaccine, if approved, stands at 2 billion doses. So as I mentioned, the one that's being talked about in the US a lot is Moderna. This was the first American company to put a vaccine into human trials. The vaccine uses mRNA to produce viral proteins. They published promising phase 1 results on July 14. At the time of that publication, phase 2 trials were ongoing, and they started phase 3 trials a couple days ago on July 27th. And the company hopes to have vaccine doses ready by early 2021. And now I will start, in the remaining of my presentation, I will talk about this vaccine. This is a report interim findings from phase 1 clinical trial of mRNA-1273 SARS-CoV-2 vaccine encoding a stabilized prefusion spike trimer S2P, and I will talk in more detail about this antigen S2P. It's important to note that product development processes that normally require years were finished in about 2 months. Vaccine development was initiated after the SARS-CoV-2 genome was posted on January 10, 2020, and the manufacture and delivery of clinical materials was completed within 45 days. And the first trial participants were vaccinated on March 16, 2020, so pretty much only 66 days after the genomic sequence of the virus was posted. The accelerated timeline generated key interim data necessary to launch advanced large-scale clinical trials within only 6 months after initial awareness of the new pandemic threat. So the way they designed their study is they conducted a phase 1 dose escalation open label clinical trial designed to determine the safety, reactogenicity, and immunogenicity of mRNA-1273. Eligible participants were healthy adults 18 to 55 years of age who received 2 injections of trial vaccine 28 days apart, so on day 1 and day 29, at a dose of 25, 100, and 250 micrograms. The vaccine was administered as a 0.5 milliliter injections in the deltoid muscle, and follow-up visits were scheduled for 7 and 14 days after each vaccination, and on days 57, 119, 209, and 394. With this report, they're showing the findings through day 57. Participants were not screened for SARS-CoV-2 infection by serology or PCR before enrollment. The trial was conducted at the Kaiser Permanente Washington Health Research Institute in Seattle and at the Emory School of Medicine in Atlanta. And like I said, they're reporting findings through day 57. So in order to understand the mechanism of this vaccine, we should take a look at the structure of COVID-19. There's its genome, the mRNA, the positive strand, approximately 30,000 nucleotide long. And the coronavirus spike protein is displayed on the virus surface over here. And its function is to aid the viral entry into the host cell. It accomplishes this by undergoing a massive rearrangement that pulls the virus and the cell membranes together and fuses them. Therefore, this protein is extremely dynamic. It changes its shape all the time. And in order to develop the vaccine as effective as possible, the researchers' goal was to stabilize it in its pre-fusion conformation versus post-fusion. So if they accomplish that task, that way the antigen will preserve its epitopes and chemistry required to elicit highest quality immune response. So more in detail about this spike protein, it has 2 subunits, S1 and S2. S1 subunit has a receptor binding domain, and this purple part is the molecular conformation, the 3D molecular shape of that receptor binding domain located on S1 subunit. And that is the domain that binds to the ACE2 receptors on the human lung cells. And it also contains this molecular glue between the virus and surface of the lung cells. And this molecular glue is what allows the SARS-CoV-2 to bind 10 to 20 times more tightly to the lung cells than, for example, SARS-CoV-1. In terms of the S2 subunit, it contains this fusion peptides or fusion loops that aid the fusion of the virus and the human cells. So a little more about this antigen that induces the response. This is an immune, this is a micrograph, electron micrograph image of the coronavirus. I thought it was pretty cool to see this bug under the microscope, the one that messed up around the entire globe. And this looks like, so this is the spike protein, and and this is the molecular 3D conformation of that protein. So the S2P antigen targeted by the researchers to develop this vaccine contains SARS-CoV-2 glycoprotein plus transmembrane anchor and an intact S1, S2 cleavage site. Here are the S1 and S2 subunits separately. And the way they accomplished stabilization of the spike protein and its pre-fusion conformation was that they substituted 2 amino acids on the top of the helix of the S2 subunit by proline, and that way they stabilized it in pre-fusion conformation form. And to be more into the nitty-gritty details of the biochemistry of it, they used the amino acids in positions 986 and 987 to substitute with proline. And after they got the antigen ready, they diluted it in normal saline and administered it as a sterile liquid for injection at a concentration of 0.5 mg per ml. So the people who received these injections, their demographic characteristics are shown in this table. There are almost equal number of male and female participants. Overall, there were 45 healthy volunteers. They were divided into 3 groups of 25, 100, and 200 microgram group. Each group had 15 participants. The median age of participants were 33 plus or minus 8.5. Majority of them, or 89% or 40 participants, were white. The next was 13% or 6 participants of Hispanic or Latino Latino volunteers. BMI median was 25.3 plus or minus 3.2. And they were followed up and reported their side effects, and the results of the side effects are shown in this table. So on the y-axis, we see the side effects, and on the x-axis, we see the percentages of the participants reporting those side effects after the first vaccination on the left side and then second vaccination on the right side. Anything in gray means mild side effects, blue is moderate, and orange is severe. So 3 participants did not receive the second vaccination, including 1 in 25 microgram group who had urticaria on both legs with onset 5 days after the vaccination, and it was concluded that this is related to the vaccine, and this participant was withdrawn from the study. But overall, there was no severe side effects that would stop the trials from moving forward. In terms of systemic side effects after the first vaccination, 5 participants, or 33%, reported mild to moderate side effects, systemic side effects in 25 microgram group, 67% or 10 participants in the 100 microgram group, and 53% or 8 participants in the 250 microgram group. However, after the second vaccination, the solicited systemic side effects were more significant. 7 out of 13 participants reported mild or moderate side effects in the 25 microgram group. All of the participants in 100 microgram group reported systemic side effects. And all of the participants, 15 out of 15 participants in 250 microgram group reported systemic side effects, with 3 of those participants, or 21%, reporting 1 or more severe systemic side effects. In terms of fever, no participant had fever after the first vaccination, and then after the second vaccination, no participant in the 25 microgram group reported fever. 40% or 6 participants in the 100 microgram group reported fever, and in 250 microgram group, 1 of the participants reported severe fever, 39.6 degrees Celsius. So in terms of local when present, they were nearly all mild or moderate, and pain at the injection site was the most common one. Across both vaccinations, solicited systemic and local side effects that occurred in more than half of the participants included fatigue, chills, headache, myalgia, and pain at the injection site. But fortunately, there were no side effects reported that would stop the researchers from moving forward and designing their protocol. And the way they did it is they used ELISA to assess for binding antibody responses against the antigen, S2P antigen, as well as against isolated receptor binding domain, the one that was located on S1 subunit. So they also assessed vaccine-induced neutralizing activity against pseudotyped virus. And pseudotyped virus is a feasible alternative manufactured by the researchers. So pretty much they use a backbone of another virus. In this case, they use lentivirus, and they attached coronavirus glycoproteins to that virus. And at the end, they created an alternative for coronavirus that would induce neutralizing activity in the body. So PSVNA stands for, it's a mouthful, pseudotype lentivirus reporter single round of infection neutralization assay. And like I said, they assess vaccine-induced neutralizing activity against the pseudotype virus. They also assess vaccine-induced neutralizing activity against wild-type live virus, and they use PRN assay for that, plaque reduction neutralization testing. So for ELISA and PSVNA, they did this assays on all participants, and they followed up for all participants, and they showed results for days 1, 15, 29, 36, 43, and 57. So pretty much all of the days that they had designed originally. However, due to time-intensive effect of PRNT, because it's dealing with a live virus, they were only able to report findings from 25 and 100 microgram dose groups on days 1 and 43. They also assessed T-cell mediated immune response in the participants induced by this vaccine, and they did it by intracellular cytokine staining assay on all participants on days 1, 29, and 43. So just as a very simplified explanation of the difference between humoral and cellular immunity. So humoral immunity secretes antibodies to find certain antigens, whereas cell-mediated immunity secretes substances called cytokines and no antibodies to attack those pathogens or to stop some processes that are developing in the body because of those pathogens. And humoral immunity is rapid or quick in their actions against antigens, while the cell-mediated immunity showed delayed through permanent action against any pathogens. So this is a pretty simplified explanation, just as a background. And so this is the results that they reported after running this assay. So on the left-hand side, we see the days. And then here are the geometric mean titers of the antibodies developed in the serums of the participants of the three groups, 25, 100, and 250. And for comparison of the participants' immune responses with those induced by SARS-CoV-2 infection, convalescent serum of specimens were also tested. So the titers of the participants are being compared to the titers of people who already had the COVID-19 infection. On the top hand side shown are the ELISA results against S2P antigen, and on the bottom it's against a receptor binding domain. So as we can see, binding antibody geometric mean titers to S2P increased rapidly after the first vaccination, with seroconversion in all participants by day 15. So look at take a look at the data from day 15. We also see dose-dependent response. So as you increase the dose, the titers increase. And there is seroconversion. And seroconversion measured by ELISA was defined as an increase by a factor of 4 or more in antibody titers over the baseline. For both assays, the median magnitude of antibody responses after the first vaccination in the 100 and 250 microgram dose groups was similar to the median magnitude in convalescent serum specimens. And in all dose groups, the median magnitude after the second vaccination, so if we take a look after any day after day 29, the titers are within the upper quartile of the convalescent serum value.
Aspram, quick question. This is pooled serum from each dose group?
What do you mean pooled serum?
Pooled. So every individual, they took an aliquot of their serum, pooled it, and then did the ELISA?
Yeah.
These are not individuals. These are pools from the 25, 100, and 250 microgram groups, respectively, yes?
Yes, so this is, so this is the median, these are the median values of the pool of that specific group of the serums. Yes, that's correct. And it shows that in the parentheses, they also show the 95% confidence interval for that mean.
Is that representing in parentheses the range of responses then?
Right, yeah.
Okay, I get it.
Yeah, Dr. Schneider, that's a 95% confidence interval. So you take a random individual, you're 95% confident that they will land in between those two values.
Okay, thank you.
Yeah, thank you for that. I was going to go over the 95% confidence interval. Yeah, as we can see, the titers after the second vaccination are within the upper, towards the upper side of the range of convalescent serums. And the patterns in receptor binding domains are pretty similar to the ones against S2P antigen. There is a dose-dependent response, seroconversion after first vaccination, and robust response after the second vaccination. So in terms of responses, so these are the PSVNA values, and this 50, ID50 means that in this dilutions, 50% of the virus was neutralized, and samples that did not neutralize at the 50% level are expressed as less than 20. So they kind of established this 10 as their baseline value, and this means no neutralization response before vaccination. And after the first vaccination, we see that the values are, again, not significant. They were detected in less than half of the participants. However, after the second vaccination, so days 36 and 43, for example, if we take a look at day 43, the PSVNA responses are significant, and the lowest median value is even higher than the median value of the convalescent serum specimens. And this, for the live virus, the baseline value at which they had detectable 80% live virus neutralization, they established that value to be 4. And they derived to these values after doing many calculations that they're not even showing in the paper. So I thought discussing it would be out of scope of today's discussion. So I will just go with 4 being the baseline value, and then after the second vaccination on day 43, we see a rapid increase of those values, which are even higher than the convalescent serum values. So overall, there is definitely neutralizing immune responses. However, after second vaccination only. After first vaccination, not so much. Like I mentioned, they also assessed T-cell mediated immune response. So again, as a background, there are two types of T cells, CD4 and CD8 type T cells. CD4 T cells are further categorized into T helper type 1 cells and T helper type 2 cells. Here they're showing the frequencies of CD4 T cells producing the indicated cytokines from 25 microgram groups on top and 100 microgram dose groups on the bottom. And these, each of these T cells, TH1 cells, secrete certain types of cytokines, and those are interferon gamma, IL-2, TNF-alpha, and TH2 specific cytokines are that they're assessing are IL-4 and IL-13. And if we take a look at this graphs, we see that after the second vaccination, the frequencies of CD4 TH1 cells that are secreting TH1 specific cytokines are increasing in 25 microgram group and more so in 100 microgram group. And I'm not going to talk about each one of these colors. I will talk about this red. If we look at these red dots, they represent any TH1 response, and we see it's significant after the second vaccination. And then if we take a look at TH2 responses, the frequencies remain pretty close to the baseline value, which means that this CD4 T cell response is strongly biased towards TH1, which is a good thing because TH1 is more favorable for antiviral purposes than TH2 response. So this is about CD4 type T cells. They also assess the frequencies of CD8 T cells producing indicated cytokines from 25 microgram groups on top and 100 microgram groups in the bottom. They're looking at the response against S1 subunit as well as against S2 subunit on the right side. And out of all of the responses, the only one that was statistically significant was in the 100 microgram dose group after the second vaccination. The other ones were pretty close to the baseline, and these couple outliers were not statistically significant. So overall, after the second vaccination, there is some T-cell mediated immune response reported in the participants. And in their discussion section, they do highlight some points that they think is important to note for this phase before moving into further phases and publishing those. First point is that the two-dose vaccine series was generally without serious toxicity. And then the mRNA 1273 vaccine was immunogenic, inducing robust binding antibody responses to both full-length S2P and receptor binding domain in all participants after the first vaccination in a time and dose-dependent fashion. Seroconversion was rapid for binding antibodies occurring within 2 weeks after the first vaccination, but pseudovirus neutralizing activity was low before the second vaccination, which supported the need for a two-dose vaccination schedule. Both binding and neutralizing antibody titers induced by the two-dose schedule were similar to those found in convalescent serum specimen. And this in turn report a follow-up of participants through day 57. They weren't able to assess durability of the vaccine. However, participants will be followed for 1 year after their second vaccination with scheduled blood collections throughout that period to characterize the humoral and cellular immunologic responses. And of the 3 doses evaluated. They moved to phase 2 with 50 and 100 microgram doses. And then after they conducted the phase 2, saw that 100 microgram was eliciting the most effective immune response, and in phase 3 trials, they will be moving forward with 100 microgram dose. It's going to involve about 30,000 volunteers, and hopefully it is, they already started, and hopefully we will see publication of those trials in a few months to the end of this year. So overall, some questions that I had about this trial was, for example, the demographic characteristics. The median age was 33. As we know, coronavirus affects elderly population more severely, and it would be nice to see how is the elderly population responding to this vaccine. Hopefully we will see that in further trials. I think that overall this is very promising. It gives us hope that basic prevention science will get us all out of this. This was a point brought up by Dr. Staudinger in one of our discussions, and I couldn't agree more. It's just there are many concerns in the public, in the general public, about the speed of this vaccine and side effects, which are all valid concerns. I think that as people involved in science in one shape or form, if we could educate ourselves and people around us about these trials, and that it's not just some magic or mysterious thing, it is actual work backed up with evidence, we would be able to reach the vaccinations required to get to that herd immunity. It's going to be a lot of work. It's going to take a lot of time, but I believe that it is possible. And with that, I will finish my talk. And if anyone has questions or comments, you're all welcome.
Any questions for our speaker?
I have one question. Could I ask? So I really liked this article, and you presented it extremely well. Do you? So we're seeing these phase trials that were started in March. I mean, this might be a good whole group sort of idea. What time frame do you think we could get something like this out? And the need for a 2-dose vaccination schedule would be a interesting public health concern.
Yeah, thank you. In terms of time frame, if you mean the time where it will be completely ready and not ready to go and be administered to the general public, I do believe from what they're saying and reading their different interviews by these researchers that it will be ready early this year, and they will be, I think, they will be ready to administer it. But however, they say that this vaccine is 70 to 80% efficient, and in order to get to herd immunity, we need at least 60 to 70% of the public to get vaccinated to be immune. And for that, ideally, we would need almost 100% of the people to get vaccinated. So I think that it will be available early this year and next year, but the long-term effects of the vaccine, I think it will take longer time. And about the 2-dose vaccination, I think they will move forward with 28 days between the 2 vaccinations, and there are many other vaccines that have that are a series of vaccinations, and I think the public would be more or less familiar with this way of administering vaccines, but I'm not sure how the results will turn out.
I'm pretty sure we're all in the same boat. well, I might continue that question with I'll let someone else continue with the next question.
Thank you, student Dr. Gunderson. Dr. Schneider, you have your hand up. Go ahead.
Aspram, thank you for a great, inspiring presentation. I like it a lot. Can you comment on the fact that there is only one mRNA naked vaccine, which we're talking about, Moderna, of course. All others are vector vaccines. So everybody who worked with RNA know how it is unstable. All right, so any information you have, how they achieved stability of that mRNA, and why they, of course, which one is better, whether it is vector vaccines, which all others are vector vaccines, or this mRNA.
Yeah, so you mean the stabilization of the mRNA or stabilization of the spike protein that is translated from the mRNA?
No, let's talk about final product. All right, so we're not talking about so there are two preparations. Let's say that Chinese, it is a vector vaccine, right? So there is a virus which will be infecting the cell, injecting that particular RNA, and will be producing protein of interest, right? So, and another one is just mRNA, naked. So, question: do they discuss how RNA gets into the myocytes? All right, so because it will not go if there is nothing is leading there. And the second one, which one is better? Which one is better? Because my concern is always about mRNA because it's a pretty unstable. You know that probably as soon as you isolated RNA, you know all of those problems. All right, so what they did to achieve stability of this preparation?
Yeah, that's an excellent question, and I never thought about it that way. And they did not address that in the study. They the only thing that they mention is this vaccine is only 70 to 80% efficient. And I was thinking about what makes it not efficient. If it goes to the cell and it produces the protein, all right, why would it be? Where is that 20 to 30% coming from? But I guess that would be it. The more you're able to stabilize that mRNA and make sure that it gets to the cell and that it makes those proteins, the more efficient the vaccine is. But I did not read anywhere information about that. That's a good topic for further research, and I will get on it.
Okay, and one more question. Of course, it's all about money. So do they estimate how much it will cost? Because Moderna got about $1 billion from NIH. And so how it will come as it will be successful and it will be marketed, how much people will pay. I know that Oxford, AstraZeneca, they estimate that it will be $3 to $4 per dose.
Okay.
So do they talk about how American people will get it and how? Because look, Dr. Salk, who invented the first polio vaccine, your parents even were not born at that time, but he's called the parent of all children of the world, because when he made it, he gave it for free. He didn't patent it. That's what I was telling you on lectures. So now we don't have this altruist anymore in the world, right? So, but it's already, they got $1 billion for that. All right, so, but I don't think we'll not stop short here in terms of what NIH gave them will be automatically free of charge vaccination. So any information you've seen how Moderna will market and what will be estimated cost for the one dose?
I don't think it's going to be completely free of charge. I believe, I'm not positive about the exact value. I believe I've seen something like $35 to $45 per dose in their website. However, it might be outdated. I didn't check lately, but I know that it's going to be free of charge to the public.
I always wonder how those European airlines flying and giving you silverware and the tickets are cost the same. Now, how the Europeans can make it for $3-4 and it goes to $50-60 estimated price. All right, we hope that ours will be better, of course. Thank you.
Hopefully.
Great presentation. Thank you.
Thank you so much.
Thank you, Dr. Schneider. Just for the audience members, in the chat, if you're able to access the chat, I put the link to Moderna, and so you can just go there and click on it. It'll take you to Moderna's website and fish around in there. Are there any other questions from our audience, student doctors?
This is maybe a formal question. I just joined this this consortium, this Zoom meeting. I'm a PsyD student.
Welcome, Mr. Gunderson. Welcome.
Thank you. I appreciate it. Is it all right if I keep up with this journal club? I'm interested in medicine and trying to involve myself in discussions about COVID-19 as much as I can.
Of course, it's acceptable, and please spread the word amongst your colleagues and friends and neighbors. This is an open forum. All intellectuals and scientifically curious individuals are welcome.
Okay, awesome. Thank you very much.
Quick comment. From my perspective, this is the journal club I've been waiting to hear for a couple of reasons. We're in a mess, all of us, and it's geeks in a lab that wear lab coats and wiggle their thumb and think that are going to get us out of this mess, and it's in the form of vaccines. And this vaccine approach is so novel and so interesting and revolutionary, and that they got on it almost immediately, and they're so far ahead of everybody else, is a tribute to virologists. As a biochemist and molecular biology PhD, I happen to know that eventually at the bench, you can trace everything back to virology, and vaccine development obviously is intimately involved. It's just a beautiful story. It's the hope that I think the world is looking for at present. So thank you, Aspram. You did such an excellent job at presenting it. And I see Dr. Schneider has his hand up. Go ahead, Dr. Schneider.
Thank you, Jeff. I just would like to have a comment for the question I always ask by friends, why they can make it so fast. It just was the first sequence, I believe, which was in February, and now we have all these vaccines. The explanation is simple. They had their vaccines which were prepared, let's say, Russians had for Ebola. Everything is developed, all doses, and so what they did, they just put their different construct. And after that, that's applied. That takes about 7 days to make it. So that's why that process of adjusting what kind of adjuvants there, everything was done previously. Therefore, now when we have it here, it goes so quickly. All right, so, and that is what my not explanation, just a piece of thought for everybody who doubts now how they can make it so quick. All right, so.
No, that's absolutely correct. I read the interview of the researchers at Moderna who's been working on the vaccine, and she said that they already worked on a coronavirus vaccine in 2012 when it had an outbreak. It was, I think, SARS-CoV-1. So you're absolutely right. They already had pretty much everything. They just needed to adjust it a little bit to make it feasible for SARS-CoV-2. That's why it's so fast.
And that underlines the importance of supporting basic science in educational environments and corporate environments, and just the importance of science in our lives. Dr. Creamer, you have a question?
I do. I've got a lot of stuff floating around in my head, and I'm trying to put it all together because I'm not a virologist. But my understanding is that this is the first of its kind in terms of this technology of using mRNA, or something about this vaccine is the first of its kind. And I wonder, I can see where that's advantageous for this COVID-19 in that it doesn't seem to mutate very much or very fast. And I don't know if that's part of the reason why they're having some success in this. But really, I guess my question comes down to, I know there's a lot of other diseases that are out there that they've tried to make vaccines for that they have not had success with. And if this is something that is successful, and I see Dr. Schneider is raising his hand, maybe he can comment on this. Going back and applying this technology to those diseases or infectious agents would be something that's now able to be done.
Aspram, would you like to respond?
Yeah, if Dr. Schneider has a comment, I would love to hear his perspective.
Dr. Schneider?
All right. First of all, we need to, and you correctly mentioned that the focus of all of these vaccinations should be Th1 type of response, cell-mediated immunity, right? So why everybody is so much talking about antibodies? Because that's what easy. Take a specimen of the blood, you measure, and in half an hour you have a result and it is reflecting, not directly reflecting, but going in that direction. So now, talking about RNA vaccination, actually the first research was started in the early 90s, right? So, and it was struggle at the moment when they synthesized a pseudo-uracil, and that's made RNA very stable. And after that, it's kind of expanded and go from that. So again, what technically we're looking at we have to focus on T-cell responses. And unlike antibody response, which very sensitive to changing sequence of proteins because it follows by changes in conformation, etc. Those T cells which see antigens, they see them as a peptides, about 11, 20 amino acids only. You can imagine immediately that from the single spike protein, you will have hundreds of peptides. Therefore, probability that mutation will result in the lack of immunity, I teach always it's a slim one because they are overlapping and responding to multiple those peptides produced in the host cell and expressed for stimulation of cell-mediated immunity. So again, antibodies, it is convenient, easy, and fast to say, which reflect, but not accurately. Most importantly, T cells. In T cells, we see small peptides, and mutation will be probably not so affecting them as B cell responses. That's my comment on it. Thank you.
Any other questions for our speaker? Okay, if not, let's reveal and thank our speaker profusely for a fantastic seminar. Appreciate everybody. Have a great day and look for announcements coming. Turn your notifications on your MKRC Canvas site, and we'll keep you abreast of what's coming next. Okay?
Jeff, I tried to go for Canvas and I'm not authorized there. I'm not in the list who can go there.
Okay,
Yeah.
I can invite you again. I've invited you. You have to accept an invitation through email. Look for it momentarily, okay? Should
All
come
right.
to you in email from MKRC Canvas. Okay, bye-bye, everybody.
Bye.
Take care.